built several years ago in order to help shepherd the next generation product for the $15 billion retinal disease category that includes wet macular degeneration, diabetic eye disease, and retinal vein occlusion. And the drug that we currently have in the clinic, it's in phase 1, is invented by the founder of the entire $15 billion space itself, the scientist who actually discovered the primary target for which every one of those $15 billion of sales of drugs is currently targeting, is the same scientist who has created a novel therapy now to address what remains unmet needs within the anti-VEGF space. And I'll walk you through what VEGF is, et cetera. VEGF, which is the molecule that he discovered. The company recently went public earlier this year through a reverse merger, and so we have our, we can pause for about 5 hours and go through this forward-looking statements slide, or we can continue. Our vision and mission at the company, again, it's a humble mission, and we're really standing on the shoulder of a giant, someone who has won the Lasker Award, which is one of the most prestigious awards in medicine and science, and who very well might be on track for a Nobel Prize in the scientific founder, Napoleon Ferrara. And as I mentioned, the primary diseases for which the drug that we're developing is targeting are devastating diseases that without therapy, these diseases lead to central blindness in people. And so really devastating effects. I mentioned the market is at least $15 billion currently. It continues to grow, and by the end of the decade, should be upwards of over 18 billion, and it's primarily dominated by 2 agents right now. The product is called TH103, and it's the first dual action product in that it's targeting not only VEGF, which has been the primary mediator over the past 20 years for treating these diseases, but also something called HSPG, which I'll walk you through a little bit later in the deck. And it's designed to address the 2 major unmet needs in the space, which are A, the treatment response itself, the robustness of the treatment response, as well as B, the durability of that response, how long the drug lasts before another administration is required. These are the 2 major unmet needs in this space. Again, as I mentioned, we're in phase 1 right now, running a clinical trial. We do have a data readout for that phase 1 clinical trial that is on track to be shared this quarter. And so by the end of this year, we are very much looking forward to sharing the data from that phase 1 clinical trial. The leadership team and the board of directors is highly experienced in developing products in this exact category and has a very strong track record over the past 20 years from an execution standpoint and from a development standpoint in that space. The drug itself, as I mentioned before, is called TH103. It has many features in common with the existing drugs. So from that standpoint, it has been significantly risk reduced. However, it's of course importantly differentiated in ways that allow it to potentially address the unmet needs that I mentioned just before. I mentioned that it's somewhat de-risked. How is that? Why is it somewhat de-risked? Well, if you look on the left of the slide here, what you can see is that it's targeting VEGF, and we know that VEGF is the key primary mediator of these diseases, of these multiple diseases that I described earlier, and it's targeting that same target. So from a target standpoint, it's risk reduced. Number 2, the structure of the molecule itself is that it's a biologic, and it's capturing that VEGF molecule in a very similar way to the existing gold standard agents. So there's a very well-established path to an FDA approval, and the administration of the drug, should the drug become approved and commercialized ultimately, fits neatly within the current streamline of physician offices. Like I mentioned before, we're currently running a phase 1 study that we'll be reading out by the end of this year. We're also just announced that we initiated a phase 1b/2 study as well, and that we are expecting a readout the second half of next year. Future development is to expand. Those two trials are both in the largest of the markets, which is the wet macular degeneration market. But we have every intention to move into the other VEGF-mediated, highly prevalent diseases as well thereafter, and those include diabetic eye disease as well as retinal vein occlusion. We do have cash and cash equivalents as of the end of the third quarter of this year of $77 million, the majority of which was derived from the reverse merger that took us public earlier this year that I mentioned earlier. So let's get into anti-VEGFs, and please ask any questions as we go along if you'd like. Lessons learned over the past 20 years. These diseases that we're describing, as I mentioned, are vision threatening. These diseases without treatment lead to blindness, invariably in patients. However, the past 20 years has completely revolutionized the prognosis for patients. They've been truly miracle drugs in a lot of ways. Every one of the successful products that has been launched targets VEGF, vascular endothelial growth factor. It is the primary mediator. What else have we learned over the past 20 years? We have learned that these drugs have become blockbuster therapies. Why? Because the market is massive. These are highly prevalent retinal diseases. And as I mentioned before, at least $15 billion in net sales globally, just on the branded market alone. However, as I mentioned earlier, there remains unmet need, major unmet need. And so there is ample opportunity for improvement over the current gold standard. What are these diseases? I'm going to give you a very high level here. The retina is where we receive light and we process images through our brain in order to be able to see. We're looking at a cross section of a normal retina in this cartoon right here, and this is the very center of the back of our eyes. It's called the macula. This depression here is called the fovea. We can actually visualize with novel technology that's been advanced over the past couple of decades. It's called OCT, and it's a form of ultrasound where we can actually, with very high resolution, non-invasively, visualize those very layers of the retina, the complicated layers of the retina, and you see that here. So this is a normal retina, and it leads to normal vision. In these diseases, what happens in particular with macular degeneration and wet macular degeneration is that the blood vessels down here that are supposed to reside posterior to the retina begin to sprout and grow into the layers of the retina. Unfortunately, these vessels are fragile. They're faulty vessels. They leak and they bleed. As you can imagine, attempting to look through blood or fluid compromises your ability to see, and of course it results in reduced visual acuity, and it can be acute. It can happen over days or a couple of weeks and be devastating for patients who were previously completely independent, driving, reading, looking at their grandchildren. Suddenly, they have central vision loss, and it compromises their ability to be independent. And what does that look like? This is sort of a pseudo image of what suddenly these patients lose is their central vision, our most important part of our vision. Now, what does that look like on the OCT that I mentioned earlier? It looks like this. Suddenly, we went from this beautiful layers of the retina, the way that it looks normally and healthily. You see fluid starting to accumulate here in these dark cavities, these hyporeflective areas on ultrasound. This is this pathologic exudation that we can visualize. And so the patient comes to the doctor, they do this non-invasive test, it takes a minute, and they can visualize it. And this is characteristic of several highly prevalent retinal diseases. And the primary cause of those new blood vessels that are sprouting and leaking and bleeding is what? Is this one molecule called vascular endothelial growth factor, or called VEGF. Now, there are numerous agents over the past 20 years that have been developed and commercialized and have been extremely successful and sell billions and billions in sales in order to what? To block VEGF, to inhibit VEGF's effect and pathology. And starting with this drug right here called bevacizumab. This is the structure of this is a monoclonal antibody. It's actually FDA approved not in ophthalmology, but in oncology. But retina specialists have repurposed it in ophthalmology. They draw it up from the vial. They take a small volume and they inject it into the eye. All of these drugs are injected intravitreally into the eye itself, parenthetically. What you can see is that there hasn't been a ton of innovation. This was the kind of parent molecule, and it's very effective, and it binds VEGF right here in this pocket. You can see that one of the innovations that came thereafter is called ranibizumab or Lucentis. It's basically this fragment right here. They turn that into a drug. Okay? These are both Genentech drugs. Both of these drugs were invented at Genentech by our scientific founder, Napoleon Ferrara. So not only did he discover VEGF itself, but he also invented the first two anti-VEGF agents. Since that time, another company that you've probably heard of called Regeneron, another major biotech company, developed aflibercept. Aflibercept is another way of tackling VEGF. And then Genentech has now launched faricimab, which takes basically the Lucentis, puts it on a monoclonal antibody stick and adds another target as well. So over the past 20 years, there's been some minimal innovation over the years. Each of these drugs that's been launched has targeted VEGF, that same target, just in slightly different ways. TH103 now, Napoleon Ferrara's next innovation, is designed to be first line, best in class in this massive space. It's massive. What does it actually look like and how has it grown since 2006 to 2024? You can see these are the major agents right now. You can see they're dominated by two drugs, faricimab and Eylea. This is the Eylea franchise, aflibercept and aflibercept HD. They represent the lion's share of this massive, close to $15 billion market. What is the problem? What is the unmet need? Well, one of the unmet needs is that there's a high percentage of patients who don't respond completely, even with these great agents. That's number one. So the treatment effect itself could be improved upon. The potency of these agents could be improved upon. Number two, these agents require frequent injections into the eye. These are mostly elderly patients, very difficult to get to the retina specialist. They require monthly visits to the retina specialist. This is tremendously onerous. Every one of the attempts at innovation in this space over the past 15 years is for what? Is to increase the length of the interval between visits to reduce that burden on patients. The current solution, so what do the doctors do? They attempt to stretch the interval. So if the interval should be monthly, they attempt to stretch the interval. They say, Mrs. Jones, I know it's hard for you to get here. Why don't you come back in 6 weeks instead of 4 weeks? What does that do? It compromises outcomes. It helps the patient become more compliant, but it compromises outcomes. And we see that if you look at the clinical trial data here, you're looking at the visual acuity improvement each month over the course of 2 years here. And you can see there's improvement of 6 to 7 letters during the first year, and then it plateaus after that. There's a bit of a ceiling effect. However, in the real world, we don't see this at all. It drops off over time because patients cannot comply outside of a clinical trial setting to a regimen that's so onerous. So there's a massive opportunity here for a drug that's going to provide durability. Napoleon Ferrara, I mentioned a few times now, I can't overstate it enough. The impact that he has had on every one of our lives, public health, both in the oncology world as well as in the ophthalmology world. Again, he's a Lasker Award winner. Many Lasker Award winners go on to win Nobel Prizes, and having invented Avastin and Lucentis and now TH103. So what is TH103? So it's addressing the two unmet needs. How does it do that? Number one, better binding to VEGF itself. Number two, it stays within the eye for a longer period of time. It's actually, the molecule itself is engineered to be sticky. It sticks within the ocular tissues and therefore should retain its effect for longer periods of time and allow for that increased interval between injections. Now, a little bit of basics on VEGF biology, high level here. VEGF is actually a family of molecules. It's VEGF-A that's actually considered to be the primary driver of all of the pathology that leads to the diseases. And VEGF-A binds to one of two what are called receptors, receptor 1 and receptor 2, on the vascular cells. They're called endothelial cells. It's well established that it binds to receptor 1 with much, much higher affinity, at least tenfold higher affinity than it binds to VEGF receptor 2. Why is that important? If you were designing a drug and the goal of the drug was to bind to the VEGF that was floating around in the eye, which receptor would you want to model your drug on? Of course, you would model it on the much higher affinity receptor. So what has Dr. Ferrara done? He's done exactly that. He's taken that exact backbone of that VEGF receptor 1. He's isolated exactly the two domains within the receptor that are responsible for binding to the VEGF ligand itself. He's isolated them. He's fused them to an FC, which creates a dimer. And here's the drug now. So you have domains 2 and 3, the two salient, most important domains from receptor 1, the high affinity receptor, and he's fused it to an FC. That's the drug. And you know what's beautiful about this drug is that this drug looks a lot like the current gold standard. The drug that last year sold $10 billion worth of sales looks very, very similar to this drug. However, there's an important distinction. First, I'll tell you what's similar. Guess what? This other drug called aflibercept that I was just alluding to also contains the second and third domain fused to an FC. So what's the difference? The difference, as you can see in the red, is that domain 3. The domain 3 from the current gold standard, the $10 billion drug from last year, is derived not from VEGF receptor 1, but actually from receptor 2. So it's a hybrid molecule. What does that mean? It means that TH103 inherently, by virtue of the fact that it's derived from the higher affinity receptor, should have better binding to VEGF. And we actually see that on crystal structure, that it binds more optimally to the VEGF molecule itself. Number 2, and very, very importantly, is that that domain 3 here, that's unique to TH103, has this unique cluster of positive charge to it that the domain 3 from Eylea, this aflibercept, lacks. Why is that important? Because there are these molecules that are negatively charged within the eye. They're called HSPGs. And so you can imagine this positive charge, this unique positive charge, has an opportunity to be anchored within the eye via that electrostatic interaction. So it creates a sticky molecule to the eye. And that's the sort of secret sauce behind this molecule and what differentiates it from the current gold standard and why it's engineered for potential superiority. These HSPGs are present throughout the layers of the retina. Everything you see in green on this cross-section of an adult human retina are HSPGs. So there's more than ample molecules for molecular anchoring of the TH103 to the retina. Okay, so in summary, better binding, longer binding. What does that look like? I'm just going to share with you a few slides on the preclinical data because it's really compelling. Every one of the experiments that Dr. Ferrara ran are head-to-head against the current gold standard, the drug that sold $10 billion last year. This is an in vitro experiment. It's showing what you see on the left here. This is the ability of the drug to prevent the VEGF-stimulated proliferation of endothelial cells, which leads to these sprouting vessels that I described earlier. And you can see that the TH103 molecule with 100% inhibits it versus Eylea, which is capped out at 80%. That's very nice, but it's an in vitro experiment. Very important to go to in vivo. Of course, the gold standard in vivo model across ophthalmology for any of these products is called the laser-induced CNV model. This is a very simple experiment. You inject the drug into a mouse eye. A day later, you apply laser spots to the retina of the eye. Those laser spots stimulate the growth of VEGF-induced vessels, and you could see the degree to which the drug is able to prevent or slow the growth of those vessels. We measure the size of those vessels a week later, and that's what they look like, this bright green spot here. So smaller is better. You can see the TH103 resulting in, statistically significantly smaller mean CNV area compared to Eylea, which is the 2 bars in pink, and the controls in gray. The reason why there are 2 bars for Eylea is because there's actually 2 doses here. This dose here is the equimolar dose to the TH103. This is the apples-to-apples comparison. This dose here is a 10-fold higher dose of the aflibercept, and you can see that the TH103 still compares favorably there. We mentioned before the drug is sticky and that there are these HSPGs. Well, do we visualize that? Do we actually see that the drug stays in the eye for longer periods of time? Very simple experiment. Let's inject the drugs into a rabbit eye and let's see if we can visualize them 2 weeks later. What you can see is that the TH103 is retained within the layers with this deeper staining here compared to the Eylea, much lighter staining 2 weeks out. Again, suggesting that the drug is staying within the eye for a longer period of time. Very nice that it's staying in the eye. It has to remain active as well. Okay, I'll skip it. I know I want to make sure we get, how are we doing on time? Okay. I want to make sure we have time for questions. He repeated the experiment. Same mouse experiment. One important distinction. Instead of administering the drugs 1 day before the laser, he administered them 2 weeks before the laser. So he was actually able to assess whether the treatment effect lasted for 3 weeks after the drug was administered. Sure enough, you can see that the TH103 retains its effect 3 weeks later, whereas the Eylea is indistinguishable more or less from the control. It lost its effect over time. Again, supporting this notion, this hypothesis that the drug is retaining its effect for a longer period of time. This is what these end up looking like, much smaller size lesions from the TH103 compared to the Eylea in the middle and the control on the right. So this is a summary. We're now in the clinic, as I mentioned before. We are treating treatment-naive wet AMD patients in our phase 1 study. Treatment-naive patients. These are patients who have never seen an injection of any of these agents before. They're freshly, newly diagnosed, and they're coming in, they're getting a single injection of TH103. We're primarily, it's first in human, looking at a range of doses, dose escalating. Because it's first in human, of course, primarily we're looking at safety, but we're also measuring pharmacokinetics of the drug so that we can get an understanding if, in fact, there are indications that the drug is staying in the eye for longer periods of time. And because these drugs do work relatively readily after injection into the eye, we can look at things like the OCT, those ultrasounds that I showed you earlier, to see if there's an effect on the fluid reduction. And we can, of course, look at visual acuity, which we have every intention to do. So those are the measures that we're looking at. Very standard kind of entry criteria for enrollment into the study. As I mentioned before, we also recently initiated a phase 1B, a phase 1b/2 study. That study is going to be looking at the drug after repeat injections, because that is the treatment regimen. It's going to be a 3 to 4 repeat monthly injections, and then an attempt to extend the dose range after that. And of course, we will be exploring the other related diseases that are VEGF mediated as well. We have broad patent coverage globally. As I mentioned before, both the board and the management team has a ton of experience, and they are tremendous, very deep domain experience over the past 20 years. Former Genentech, iBio, iTech, many of the companies that were involved in the development of all the drugs that have led to the development of the category. Here's the summary slide. I'll pause there because we have a few minutes for questions, and I'm happy to take any questions.
Would anyone like to ask a question? Okay, I would like to just...
Yes, please.
You went through the structure of the drug and the difference in efficacy. In terms of market share, any expectations or head-to-head efficacy compared with the drug, the dosing interval?
So in terms of what we would like to see in terms of an extended dosing interval. Excellent question, Robert. So the current drugs, it's a very heterogeneous disease. There are patients who have very aggressive lesions, and you have patients who have more indolent lesions. And so some patients require literally monthly injections, and other patients can get away with either bi-monthly or every 3-month or every 4-month injections. So you can create like a bell curve of what percentage of patients currently fit into each one of those categories. Because of the nature of the biology and the complexity of these lesions, a one-size-fits-all is highly unlikely, and I think is biologically implausible. What we're looking to do, and what would be a major advantage to this space, is to shift that bell curve to the right. So you have higher percentages of patients with longer durability. So you have higher percentages out at that Q4 month, Q3 month level, and lower percentages of patients who require the monthly injections. And so that's that would be a major advantage to the market.
With more than 6,000 small and micro cap companies listed, if you're looking for the next Apple, then Channel Check truly is the orchard. It's an investor community where you can access independent Wall Street research, advanced market data, balanced news, corporate videos, and podcasts. You can even register to attend in-person Meet the Management Series luncheons, and to be part of the preeminent annual Small and Microcap Investor Conference, NobleCon. And here's the best news. Since Channel Check is sponsored by the featured companies, premium membership requires no investment. It's absolutely free. So check it out. Log in today. Find out why Channel Check is North America's leading source of research on the companies that just may change the world.